Hello everyone. So my research is over program translational frameshifts, and I study this in a bacteriophage called BryanRecycles. Bacteriophages or phages are just viruses that infect bacteria. A program translational frameshift is a phenomenon in where two peptide products or proteins are produced from one mRNA sequence. One mRNA transcript. Normally, just one protein is produced from one transcript. So there is a confirmed frameshift in BryanRecycles in the gene that encodes for the tail assembly chaperone, or the TAC gene. There is a predicted frameshift in the DNA primase gene. How can we confirm whether or not there is a frameshift or not? So we'll design and use a wet lab procedure to experimentally show the frameshift that is in the TAC gene. We'll then use the same procedure to test for a frameshift in the DNA primase, ultimately using TAC as a positive control. So our methods: we'll isolate and purify our target DNA, insert that into a plasmid, which E. coli will then uptake and then produce our protein of interest. Protein of interest will be isolated and purified for further analysis. This is bioinformatic identification, so we just use computer software to predict and find possible frameshifts. Here are wet lab results. These two gels show that the PCR products indicate that we have the correct DNA sequence that we are looking for. And so from that DNA, we can create the proteins. The protein concentrations are high enough yield to continue with analysis. So SDS-Page. SDS-Page is a gel electrophoresis for proteins that separates them using their molecular weights. The protein ladder here spans from 250 kilodaltons down to 10 kilodaltons. The results for TAC, there should be two bands at 11 and 37. Since we know there is a frameshift, there should definitely be bands here, especially since the protein concentration is high enough. For DNA primase, there should be one at 16 and 35. Fortunately, there are absolutely nothing here for DNA primase, which means negative results. For TAC, there's about 7 bands, which is inconclusive. There should only be 2. Our prediction is that sample runoff ran from this well into this one and contaminated that way. So next steps will be to continue with SDS-Page until we get a good enough result, and then go on to the chemiluminescent immunoassay, which will further confirm that we have the correct products. We'll sequence the proteins, and that will ultimately confirm whether a frameshift is present or not. This experimental design, if it proves successful, we will be able to use this on other phages that have predicted frameshifts and be able to confirm the presence of a frameshift via wet lab procedures. Thank you.